said in the last lecture, when I spent a lot of time on that very first slide, it was a cartoon diagram of cardiac myocyte, and I went through a lot of detail on what ion currents affect the action potential and how and when. And so this is a compact summary of the things that I said in that slide, and it just illustrates for you when different currents play a role. It shows you the kinetic behaviors of this current and the phase of the action potential during which they contribute in the way they do. So this is something that you can do. I left you on this slide. We were discussing modern approaches to modeling voltage-gated ion channels. And I had presented to you a multi-state model of potassium current known as the transient outlet current. And the hypothesis is in this model is that the current is, its behavior is described using a complement of states. This is the activation pathway for that particular potassium current. There are 4 closed states leading to an open state, and then there are 5 inactivated states. And so the question was, where do the equations of evolution come from that describe the behavior of this kinetic model of an ion channel? And so we derived the expressions for the time rate of change of occupancy probability for the various states in such a channel model. And that was this family of ordinary differential equations given there, 1 for each state. And then we went through the equation that gives the current through a channel or a complement of channels when a certain fraction of them are in the open state. And so we need this current expression ultimately to integrate this channel model into a model of the myocyte and use the current through this complement of channels to describe how memory potential of that model changes over time. We'll come to that in a minute. We talked about the form of the transition rates, the rate at which one switches between different states in this model. It has this exponential form, so we're going to talk more about this now. And I did not mention a constraint on this microscopic reversibility. There is an additional constraint that says in this model a channel must be in one of these 10 states, and so these occupancy probabilities always sum to 1, and therefore we don't need to numerically integrate all 10 of these equations. We can integrate 9 of them, and the final unknown occupancy probability is just 1 minus the sum of the other occupancy probabilities. So that is a constraint. But I want to talk about microscopic reversibility and why that is important in this particular channel model, since it has what we'll call loops, these pathways by which one can transition, for example, from C1 to C2 to C2 to, or CI2 to CI1 and back. So this is what I'm going to call a loop, and you can traverse that loop between this cycle of states in this direction or in this direction. If the channel model only had this set of linear states, if one progresses from C0, C1, C2, C3 to the open state, there are no loops in such a model. But there are loops in this model. And so what does it mean that there are loops energetically? And so it means something very important from statistical mechanics, and in particular a theory known as Eyring rate theory. That theory describes the rate at which a chemical system transitions from one state to another under the assumption that that transition involves traversing an energy barrier. And so let's think of these 3 states that do form a loop. We can transition between these states in the indicated manner. And this is a plot of the Gibbs free energy associated with being in state 1, state 2, and state 3. And it illustrates the amplitude of the energy barriers that are involved, that must be surpassed in transitioning from state 1 to state 2, for example. And statistical mechanical theory, in particular Eyring rate theory, tells us that the rate of transition from a state of Gibbs free energy G1 to a state of Gibbs free energy G2, where the energy barrier separating them is G1 star minus G1. That rate in transitions per unit time is KT over H, not the cube, that individual rate is KT over H times this quantity here. And so this exponential term here is the height of this energy barrier. So this times KT over H is that transition rate from state 1 to state 2. So what we're going to do is to form, in this equation here, the product of the transition rates moving in this direction around this loop. That is from state 1 to state 2 to state 3 and back to state 1. And so the transition rate from which we go to 1 to 2 is given by KT over H times this term, state 2 to state 3, it's given by this term, state 3 back to state 1, it's given by this term. And so if we form that product, this is the expression that we get. So let's just hold this thought for a moment and do the same calculation, but this time going in a different direction around, not the clockwise direction, the counterclockwise direction. And so now we're going to go from state 1 to state 3 to state 2 back to state 1. And if you form the product of those transition rates, do the calculation, it's the exact same number. All this is saying intuitively is if you begin in one energy state, you progress around the loop, and you return to that same state. You must return to that same energy state. If you don't, you've either gained or lost energy. And so microscopic reversibility is saying that there's no energy loss or no energy gain transitioning around the loop, then this relationship will be satisfied. Why is this important? Because if we call this rate K12 and this rate K23 and so forth, and we form the product of those rates traversing the loop in the clockwise and set it equal to the product of those rates traversing the loop in the counterclockwise direction, we can solve for one of the transition rates in terms of the others. And so microscopic reversibility is allowing us to constrain one of the unknown, for each loop, one of the unknown parameters. In our model, we have 4 loops, and so we're going to apply this principle of microscopic reversibility 4 times, and thereby obtain 4 constraints on unknown transition rates K. So it means that microscopic reversibility reduces the parameter space of the model and helps constrain the model and enable us to have to fit the behavior of the model to real experimental data by varying fewer parameters, because some of them are constrained by virtue of the principle of microscopic reversibility. Okay. So just to summarize the new thing I added here and what we talked about in last lecture, what exactly do you need to know from the last lecture and this little snippet here? Well, you need to understand what currents are at play during different times of the cardiac action potential. And you need to understand qualitatively the kinetic behavior of those currents. Kinetic behavior means you know the sodium inward current, which starts the upstroke of the action potential. It's a very fast, transient current. It's an inward current. And you know a little bit about the calcium current, because we talked about that last time. And you know a little bit about the potassium currents, because we described when and how they play a role in polarization and that they are outward currents. So familiarize yourself with those. We talked about a different way of representing the Hodgkin-Huxley sodium channel model, one that used a multi-state model. And based on that kind of multi-state representation in which channels undergo a sequence of physical conformational changes during the gating process with the membrane potential that is imposed across the cell membrane, channel transitions through different conformational states. We have a representation of those states in our channel models. And you need to know how to derive the differential equations that can be solved for occupancy probabilities of different states in a channel. I might give you a three-state model. And I might say, write down the 3 ODEs for the time rate of change of the occupancy probabilities for states 1, 2, and 3. So you know how to do it from the slide I just showed. It's essentially laws of mass action. So to do it, you need to make an assumption that I'm in this state. What are the rates at which I enter that state from connected states, and what is the net rate at which I leave that state? That kind of information is how you can write down these governing ordinary differential equations, and understand the constraints that can be placed on these kinds of channel models. And there are two broad types of constraints. The first is the occupancy probability must sum to 1 across all of the states. You've got to be in one of them. There are no others. The second constraint arises when a channel model has loops. And there, that constraint is microscopic reversibility. So know what that constraint means thermodynamically and how to apply it. All right, so we've spent time talking about voltage-gated ion channels, a different way of representing them. I now want to give an example of the ways in which modeling of membrane transporters has been approached. It's very similar to what we just went through with ion channels, but with a few truly simplified assumptions. And so while the slide that I will show you on the modeling part is a little bit daunting and dense, conceptually it's quite simple. So we're going to talk about a recent model of the sodium-potassium pump. You know that the job of this pump is to extrude sodium that enters during the action potential. You know the stoichiometry of this pump. It's 3 sodiums go out at the cost of importing 2 potassiums. Now, this is a relatively high consumer of ATP in the cardiac myocyte. Because of that stoichiometry, per cycle, it generates a net outward movement of charge. 3 sodiums go out, 2 potassium comes in, so that's a net outward movement of charge. And because of that, even though it's not an ion channel per se, it's not a voltage-gated ion channel, it is a membrane transporter that generates a current. And therefore, if we were to change the extent to which this particular protein is expressed in a cardiac myocyte, to upregulate it or downregulate it, that does affect a transmembrane current. It will have an effect on the nature of the action potential, which you can intuitively predict from the fact that this transporter is generating a net outward current. So if a cell is starting to express more of these transporters for whatever reason, the outward current through them is going to be larger, and that will have some, maybe a small, maybe not, effect on the action potential, depending on how large this dysregulation is. And so here's the dense slide that is really a simple slide. And we're going to use this to illustrate approaches to modeling membrane transporters that are commonly used in the literature. So again, this is the sodium-potassium pump, and here is a 4-state model of that pump. So let me tell you what the states are. So this part of the diagram is inside the cell. This part of the diagram is outside the cell. So let's begin here in this state where E represents, in the nomenclature of the paper that I took this model from, E represents enzyme. It's representing the pump protein complex. P is a phosphate bound to the pump, and 3 and A represent 3 sodium ions that are bound to this transporter complex. So when these 3 sodium ions bind to the pump, and they're binding when these binding sites are exposed on the inner surface of the membrane, so these are intracellular sodium ions that are binding to to this transporter. The transporter undergoes a conformational change in which the sodium is translocated across the membrane and entering what I'm calling here state 2. And for state 2, I've denoted state 2 as the enzyme or the pump complex bound to a phosphate, but now bound to 2 potassium. So what happened to the sodiums? And so they've dissociated, they've unbound, they're released into the extracellular environment. That's the function of this pump. And in their place, 2 potassiums have bound. But this model makes an assumption, an important assumption. And this kind of assumption is known as a rapid equilibrium approximation, or a rapid equilibrium assumption. That phrase is going to come up a lot in this lecture and the next lecture. What does it mean? It means that when the pump undergoes the conformational change that translocates these 3 sodium ions across the membrane, the rapid equilibrium approximation that is being made here is that instantaneously those 3 sodiums are gone from the pump. They unbind. And instantaneously 2 potassium ions jump onto that pump. And so the model doesn't explicitly describe the process of sodium unbinding and then potassium binding. The model assumes it all just happens instantaneously. We're not going to bother with the kinetics. It happens so fast. These other kinetic processes are what are controlling the dynamics of the system. So we're just going to ignore the details of that unbinding of sodium and binding of potassium. Okay, so we now have the pump bound with 2 potassium ions. This step here reflects a binding of ATP to the pump to provide energy for the translocation of potassium ions back across the membrane. And again, a rapid equilibrium approximation is made here. Those potassium ions just unbind instantaneously. 3 new sodium ions jump onto the pump. And we're ready to repeat this cycle. So these are the states where this rapid equilibrium approximation is made. But I think this is otherwise an intuitive diagram, and you get the meaning of what it means for this pump to cycle and transport sodium and potassium. And so how would you begin to model this dynamical system? You would use the same thinking that we did for ion channel models. Let's define an occupancy probability for each one of these 4 states. And then we use exactly the same logic, laws of mass action, to write the dynamical equations for the time rate of change of occupying state 1. And if you go through these equations, they are developed using exactly the same logic as we did for the ion channel model. And so you write one of these equations for each of the 4 states. But in most modern action potential models, the action of these pumps is not modeled kinetically. And that's because the cycling is assumed to, these kinetic steps are assumed to occur so fast that for a given sodium and potassium concentration inside and outside the cell, if we were to look at the states of many different sodium-potassium exchanger molecules, the distribution of those states, the occupancy probabilities, will have achieved their steady-state values. So what does that mean in terms of these equations? In terms of these equations, we're not going to model the kinetics with which these occupancy probabilities change. We're going to assume that they take on their steady-state values immediately. That's another kind of rapid equilibrium approximation. So the left-hand side just becomes 0. Reach their steady-state values instantaneously. And so by setting this side to 0, we now have 4 algebraic equations in 4 unknowns. And the 4 unknowns are the state occupancy probabilities. And armed with those, and so we can solve for those probabilities. Now, it can be messy, but we use Mathematica or Maple or something to solve these four equations for the p's, and we get the answers, we get the algebraic expressions. And so armed with the values of the p's, we can now get the relevant fluxes through the pump. So what's going to be the net outward sodium flux? This is where sodium transport occurs. And so the net outward sodium flux is the probability that the steady state probability that we're in occupying state 1 times the transport rate A2 plus, which is this rate here. So that's one way. So that represents an outward flux of sodium minus the probability that we're in state 2 times the reverse transport rate, which is A2 minus. So the net flux through this pathway is the net sodium flux through the pump. And if we know, and we've already solved for P1 through P4, so we've got an equation for that net flux. And scaled by 3 because this pump is transporting 3 sodium ions each cycle. And we follow the same logic for potassium. We can get the net potassium flux through the pump by looking at this pathway, that's the potassium transport pathway, and going through the same logic. So we can write down these equations. And so in a computational model of the cardiac myocyte, we want to know the sodium, the flux of sodium contributed by this pump, because we want to track how sodium ion concentration is changing in the cell. We're going to use this equation here, which tells us the net rate at which sodium is transported out of the cell by that pump. So is that clear? Are there any questions about how we reach that conclusion? Yes?
So we assume that the ion that usually binds to the thing only binds to the transporter once it's already on the correct side, or that said that it was ended.
So what we're assuming is that for 2 of these states, this one and that one, that as soon as sodium is moved across the membranes in the outside face of the membrane, it instantaneously jumps off, and potassium instantaneously binds and is then available to be translocated in that state.
So only once it's outside.
Yes, yes. The theory is really no different from that which we developed for ion channels. So what do you need to know about that? We will be much more quantitative about what a rapid equilibrium approximation means shortly. But I've defined it for you. I've given you some examples, so at least at this point know it intuitively. Understand that when we typically think about transporter molecules and how they function, the operational equations, the equations that we use in models of the cardiac action potential represent steady state fluxes through these transporters. And so we've worked through how those fluxes are derived. So you should understand that quantitatively and intuitively in that prior example, because I might give you an artificially constructed pump model and ask you to do these kinds of analyses. All right, so we've talked about voltage-gated ion channels as one kind of component that goes into a model of the action potential. Membrane transporters as another kind of component. Here's another aspect of models of the action potential that has to do with compartments and ion concentrations. And so in my example here, I'm only talking about calcium, but we care about the concentration of at least calcium and sodium and potassium in the myocyte. And depending on the model, we may care about concentrations of a whole range of metabolites and signaling molecules, but
The models that we're going to talk about here, calcium and sodium and potassium are the big ones. And so here is a cartoon diagram of a model of a cardiac myocyte with regards to the different calcium compartments that are in the model. And so the first that we think of is this big compartment, which represents the bulk cytosol of the cell. And so virtually every model of the myocyte makes the assumption that these cells are sufficiently compact that membrane potential and ion concentrations in the cytosol are equilibrating quickly and changing everywhere in the cell at the same in the same time, or changing in this everywhere in the same compartment at the same time. So that assumption is made within the cytosol. Calcium is equilibrated throughout the cell, and however it's changing in time, it's changing uniformly in time everywhere within the cytosol of the cell. There are three other calcium compartments shown here. One is the network SR, which we described last time as a organelle. SR is an organelle, it sequesters calcium. Network SR extends throughout the cell. It has circupumps in its membrane, and they transport calcium into this organelle. That calcium then diffuses into the junctional SR, those localized regions of SR that form the dyads. This is what we talked about last Monday. And so network SR and JSR will be two separate calcium compartments. And then we talked about the dyad itself, and in this kind of model, we're taking all of the dyads in the cardiac myocyte. We're lumping them together. We're going to call them the subspace. It is the space that is equal to the summed volume of all these tiny, tiny dyadic spaces, tens of thousands of these dyads in the cell. We're all lumping them into one, and that is this compartment. And so this schematic model of calcium handling in cardiac myocyte has four calcium compartments. So we care about how calcium changes in time within each of these compartments. It's an important, critically important dynamic variable in these models. So how do we analyze this? Well, the time rate of change of concentration of some ion, an ion species, the time rate of change of that ion species within a compartment is equal to the net, so I is a current, this is the net current carried by that ion into that compartment. The negative sign here just denotes the fact that whatever the compartment is, to compute the time rate of change of ion I in that compartment, we've got to compute the net inward current associated with ion I into that compartment. So we care about how much calcium is directed into the subspace, or how much calcium is directed into the JSR. The net current, the sum of all the inward and the outward currents. It's got to be taken in the direction that is inward. And then we divide that quantity by these terms. Z is the valence, F is Faraday's constant, and V is the volume of the compartment. Now, this makes intuitive sense to me, because if you change the volume, if you make the volume smaller of a compartment, a given inward current, or a given amount of charge moving into that compartment is going to have a much larger effect on the rate at which concentration changes in that compartment. So it's a smaller compartment. So things are going to change more quickly. The volume gets bigger, that equivalent current, that equivalent amount of charge will produce a less rapid change. So V being in the denominator here makes sense, and current being the numerator makes sense. The more current, the more rapidly concentration will change in that compartment. So this is a general form of an equation for the time rate of change of ion concentration in any compartment within a cell. And so we've talked about all of the essential ingredients that are part of every computational model of a cardiac myocyte these days. And so, well, I guess what I haven't talked about yet is this equation for the time rate of change of membrane potential. So the membrane potential, the time rate of change of membrane potential is going to be proportional to the net inward flux of charge into the cell. So the cell membrane has a capacitance that is determined by the surface area of the cell. The capacitance of a bilayer lipid membrane is considered to be a biological constant. It's about 1 microfarad per square centimeter. So if you know, excuse me, the volume or the surface area of the cell, and you compute the capacitance of the cell membrane, and we're assuming the cell is small enough to be isopotential here. And so the time rate of change of membrane potential is just equal to the sum of all of the currents directed into the cell, scaled by 1 over the capacitance. So we then have the family of equations that arise from modeling the voltage-gated ion channels that are present in the cell. We have the complement of equations that arise from modeling time-varying ion concentrations in the cell. We have equations which we've already worked through, the form of which we've already worked through, that give current through a voltage-gated ion channel when it's in the open state. And that current depends on the ion concentrations to which that channel is permeable through the Nernst equilibrium potential. And then we have currents through the various membrane transporters, and we work through the algebraic equations, the formulation of algebraic equations that describe those currents. So these are the basic parts of every computational model of a myocyte. So if you count up the number of states in modern models, the number of ordinary differential equations in modern models, it's like 100, 150, 200, varies from model to model. Models are becoming increasingly high dimensional. So what does it mean to simulate a model of cardiac action potential? Well, you've got to take all of these ordinary differential equations and current equations, and you've got to numerically integrate them in time. So you have to solve for channel occupancy probabilities, and you have to solve for concentrations in different compartments. You have to compute currents to get the time rate of change of membrane potential, and you're numerically integrating that as well. And so you will compute V of T. And so now you can use that kind of model to study how a perturbation of any of these different factors in the model affects the action potential, or affects a particular membrane, or affects a particular ion concentration, or how different components interact with one another. This is how you do it. This is the nature of the model. They're vast systems of coupled nonlinear ordinary differential equations that are numerically integrated in time to simulate the behavior of the myocyte. So that's the core of modern myocyte models. And so in the title of my last presentation, 50 years of cardiac myocyte modeling, it's now 55 years. I think it's fair to say that of all mammalian cells, cardiac myocytes, to me, are the most quantitatively well understood cell in mammals. Because there is a 55-year history of building thoughtful, experimentally based models of these cells and trying to understand how they work quantitatively. And so these models have been applied in many, many, many different ways. And I'm only going to show you a few here. And it's remarkably surprising the degree to which these models have provided significant biological and clinically relevant insights. So much so that I think it's a true statement that if you're a person in the world of cardiac biology, studying ion channels or studying properties of the action potential, and you may be an experimentalist, maybe not a biomedical engineer, but it's become completely accepted in the field that what you do must be interpreted through the use of a model. And so people who study ion channels, they're not trained like you necessarily, they're not biomedical engineers, but part of the field now is if you're going to talk about an ion channel, you better talk about a model of that ion channel, not just the data. And so that is the case in our field. Modeling has seeped thoroughly through the biological world of studying the heart. So I'd like to give you a couple of examples of the impact, the real impact of modeling. And I want to begin by talking about work that we did years ago in thinking about heart failure and the cellular phenotype of heart failure and the basis of arrhythmias in heart failure. So, heart failure is a major cardiovascular disease. It is one of the leading causes of sudden cardiac death. Sudden cardiac death meaning a sudden unanticipated arrhythmic event resulting in death. And so there's been a great deal of concern about what is the mechanism of sudden death from heart failure, and what is going wrong with the action potential in heart failure, and why is it a problem? Well, if you isolate individual cells from the failing human heart, which you can do because people have heart transplant surgeries and those explanted hearts can be made available for scientific use. And so there's a vast literature on the electrophysiology of cells, of human ventricular myocytes that have been isolated from these explanted hearts. And the universal signature of heart failure in terms of how a myocyte functions, there are two parts of it. The first part of it is, and I'll show you in a minute, an example of this. The action potential is tremendously prolonged. It can be double what it is normally. So normally in humans, action potential duration at a pacing frequency of 1 hertz can be 350, 400 milliseconds. If you pace a failing human ventricular myocyte at 1 hertz, you can see action potentials that are twice as long. So action potential duration is severely prolonged. And that has very important consequences for the genesis of arrhythmia. And we will talk in detail about why that is. But action potential prolongation promotes a form of arrhythmia that is known as an early after depolarization. This is an abnormal repolarization of the action potential that occurs very late in phase 3, early phase 4, just when normally the action potential would begin to drop in amplitude and repolarize. And early after depolarization is an abnormal blip on the action potential, an abnormal depolarization event that is believed to have the potential of triggering arrhythmias in the whole heart. These become more likely when action potential duration is prolonged, and so prolongation is a serious issue. The second universal signature of heart failure is a very much reduced calcium transient. If you look at the magnitude of the calcium transient, the failing myocyte could be half that in a normal myocyte. The heart is contracting less strongly. The signal driving contraction is stronger. And you see this in myocytes isolated from patients. And you see this in every animal model of heart failure that's ever been studied. So these are two universal features of heart failure. So a lot of attention began to be focused in the late 1990s and early 2000s on what causes these changes of the action potential and the nature of the myocyte. What's happening? And so this is at a time when gene expression microarrays were just being developed, and it was just becoming possible to do large-scale assay of the number of messenger RNAs that were being produced that encode a particular type of ion channel or membrane transport. And so that was one direction of research. Another direction of research was to not necessarily use high-throughput genetic assays, but to measure the expression levels of particular messenger RNAs for very particular proteins. Another was to patch clamp record from individual protein channels and patch clamp studies and estimate the density of these channels in cell memory. So many, many different directions of probing the myocyte to determine how the expression of certain ion channels and membrane transporters was altered in heart failure began to converge on 4 simple observations. And these are the 4 observations. So we've talked about this transient outward current. In human heart failure and animal models of heart failure, it was seen that that current was downregulated. There are less of these proteins produced. There are less of these channels available. Maybe 60% or so fewer than in the normal myocardium. We've talked about the membrane current IK1, that is the current that turns on late in the action potential to finally repolarize the cardiac action potential. It's a potassium current, outward potassium current. That was downregulated by 30%. 2 other findings. I'm calling it ATP2A, that's the gene name. This is the circa pump. It was downregulated by 50% or 60%. It's this pump here. And so that's the pump that transports calcium into the SR. So there's fewer of them. And then the sodium calcium exchanger pump, you know that helps extrude calcium from the cell during diastole. That was actually upregulated. So this picture began to emerge that these 4 proteins we know play a very, we understand their role in the cardiac myocyte, but their expression is altered in heart failure. And at the time, the interpretation of these data was the following. And this interpretation comes from what I like to refer to as a mental model. So people looking at these data said, we want to explain action potential duration prolongation. Look, there are 2 potassium currents that are downregulated. Potassium currents are outward. If there is less potassium outward current, I guess that explains action potential prolongation. It makes intuitive sense. I see the logic. Turns out it's completely wrong. But that was the view at the time. The view about these players was, oh well, I can understand how that might affect the calcium phenotype in heart failure, a smaller calcium transient. If there's less circa pump expressed, you imagine there's less calcium transported into the SR. Remember we said that's the major source of calcium release during the calcium transit in the action potential. So there's less calcium in SR. It makes sense that maybe that is helping to explain why the calcium transient is so blunted. And increasing NCX would only emphasize that, because this sodium calcium exchanger extrudes calcium from the cell during diastole, so it's going to be another factor driving calcium out of the cell, preventing it from being pumped into the SR. So these kind of made sense in terms of the calcium phenotype, and everybody thought this made sense in terms of the action potential phenotype. So we came along and decided, let's make this quantitative, let's build a model of the canine ventricular myocyte, and let's make a model of what that cell looks like normally, and let's make a model of what it looks like in a canine model of heart failure. Let's build in these mechanisms, and let's see if this view is correct. And so that's what we did. We constructed this model. Here's the data, experimental data, that I referred to when I was talking about the previous slide. So here's a normal action potential measured in the dog heart. It's just a single cell, a single action potential recording. Here it is in a model from an animal with a failing heart, very much prolonged. Here are the changes in the calcium transient, big calcium transient in the normal myocardium, very blunted one in the failing myocardium. And so what we decided to do was, okay, we've got these 4 changes, we know what they are, let's impose them in the model. How do we downregulate the transient outward current in the model? We're just going to dial in a smaller conductance. There's fewer of those channels, so we'll just reduce the conductance associated with those channels, and we'll reduce it by the measured amount, which is like 67% or so. So we reduced the amount of transient outward current in the normal cell model. Remember the red here is the normal action potential. Yellow here is what we get when we dial down the transient outward current, because that's getting downregulated by that. Action potential duration doesn't get longer. In fact, it gets shorter. That in and of itself was non-intuitive and anomalous. But forgetting that one point, what we can say is that that downregulation alone certainly doesn't account for this action potential prolongation. So let's keep going here. Let's layer onto that downregulation of the next potassium current, IK1. Let's turn that down by the prescribed amount. That's shown on the blue trace. Well, the action potential got a little longer, but it doesn't get anywhere close to capturing this cellular phenotype. So we were at a place here where current understanding doesn't seem to be borne out in a quantitative model. It's not predicting what we think is our mentally intuitive phenotype. In fact, it's not working. So then we continued. In addition to these changes, we downregulated the circuit pump. And boom. Action potential duration is dramatically prolonged. In fact, that's pointing to the wrong thing. It's this curve right here. And that's the punchline here. We then upregulated NCX, and that attenuated action potential a little bit, but it's still prolonged, and clearly the key thing is downregulation of the circuit pump. And so people scratch their heads and wonder, why in the world would downregulation of a calcium pump have this whopping big effect on action potential duration? But not the potassium currents. We thought they were the players, not the potassium pump. So we went on to probe this. And so the answer lies in some important properties of the L-type calcium channel that were really beautifully explored by David Yu in our department. And I'll summarize these properties in this really, whoops, the animation didn't work. Okay, so let me walk you through this. So L-type calcium channels have the property that not only do they undergo voltage-dependent inactivation, like sodium channels, they also undergo what is known as calcium-dependent inactivation. We refer to that as CDI. And that means that when the calcium channel, remember it's facing into the dyad, this is a little tiny space here, 15 nanometers away from the cell membrane, is the JSR membrane with the rhianidine receptors. When these calcium channels open and calcium flows into the cell, since this is such a tiny space, that flux of calcium creates a huge change in calcium concentration in that compartment. And that calcium then binds to a molecule called calmodulin, which is tethered to the L-type calcium channel. This is one of David's contributions. And binding of calcium to calmodulin then induces inactivation of the calcium channel. Inactivation meaning that the channel will tend to transition to states from which it can't open. And so that is a negative feedback effect on the L-type calcium chain. That rapid rise of calcium, which is particularly rapid when these rhianidine receptors open, because they're releasing a lot of calcium. The release flux is bigger than the trigger flux. And so logic would say that released calcium must be really powerful in inactivating these calcium channels. In other words, promoting them to gate into states where they can no longer conduct. And so calcium channels are undergoing two kinds of inactivation at once. An intrinsic inactivation due to the presence of an electric field across the membrane, it's changing membrane potential, like sodium channels do. And then they're getting shut off by the calcium signal in this compartment. And it's a negative feedback effect. And so what's the result of that negative feedback effect? Well, it attenuates the inward flux of calcium through these channels. If it's causing them to inactivate, few of them are open. And so the inward triggering calcium current is getting small. This is one way in which the cell acts to abbreviate that trigger current. There's no need to bring in all that calcium if you've already triggered release from the SR, so let's attenuate it. Calcium-dependent inactivation does that. Now, what would happen if the amount of calcium released from the JSR through these rhianidine receptors was reduced? And what we're speculating here is that when that circuit pump is downregulated in heart failure, there's less calcium in the SR, there's less calcium available for release. What would be the effect of that? There would be less inactivation of the calcium channel than occurs normally. And so the inward calcium current wouldn't get attenuated as it does normally. It would be enhanced. And maybe it's that enhanced inward calcium current that's driving action potential prolongation. And that, I won't take you through all of the modeling results that show you that. That is the case. This is the mechanism by which that dysregulation of a calcium pump feeds back onto a membrane current that has a very important role in shaping action potential duration. If you just downregulate the pump, you produce varying degrees of action potential prolongation. If you go do an experiment where you pour in a drug that blocks that pump, you get action potential prolongation. And if you do experiments to measure the amount of calcium that is in the SR when that pump is dysregulated in heart failure, you see it's close to that. It's reduced from normal. It's close to what we measure and predict in the model. And so this is a prediction that has stood the test of time. And I think it's a really nice illustration of how sometimes our mental models fail us dismally. Our intuitive understanding of what role downregulation of these potassium currents would play is just completely wrong in this case. So wrong that it couldn't explain the basic action potential phenotype of the failing myocyte. And there was something else going on. There's something else was kind of buried and strongly nonlinear, kind of mysterious, but it came out through a model. This was a prediction made by the model. It was subsequently validated very elegantly when David Yu ablated the calcium binding sites on calmodulin genetically and produced these animals in which this regulation was, these myocytes in which this regulation was lacking, and showed that tremendously prolonged the action potentials in these cells. And so now there are many efforts in some clinical trials underway to deliver circuit pumps through viral vectors, genetic engineering to treat heart failure, gene delivery to treat heart failure. In fact, being led by the work of a graduate from this biomedical engineering program, Roger Hajar. Okay, how are we doing on time? So now let's go to another class of problems that have been tackled with models. I'm going to show you one example of it. There are just hundreds of examples of this kind of work. And so in this work, what these investigators did, Colleen Clancy and Yoram Rudy, was they used a model of the cardiac action potential, and they developed a new, different model of the sodium current. The details of that model aren't important for us. It's not something that you have to know. This is the model of a normal sodium current that they used. The sodium channel would transit through 3 closed states to an open state, and then to 2 inactivated states hanging off here. Now, I should say that there's really, you've got to take the structure of these models with a grain of salt, because there's not a lot to guide one in the formulation of how these states interconnect with one another. And so this is just one hypothesis, one model that can account for properties of cardiac sodium currents. Other models with different structures can as well. the important thing that they did was to use data on the nature of the sodium current recorded from myocytes in patients with a genetic disorder known as long QT syndrome. And so long QT syndrome is a complex genetic disorder that is multifactorial. Long QT means that the QT interval of the ECG is prolonged. That reflects prolongation of action potential duration. These patients are at higher risk for serious arrhythmias. And so understanding the nature of long QT syndrome and its treatment is important. And so these mutations that underlie long QT syndrome can affect sodium channels, potassium channels, and calcium channels. They were interested in a particular mutation known as the delta KPQ mutation that affects the cardiac sodium channel. And so what they did was to take electrophysiological data in a literature in which these defective channels had been expressed in host cells, and very detailed electrophysiological recordings had been done of channel properties. And they formulated a model of sodium channel with this delta KPQ mutation. And the essence of the model was to introduce a new set of states below the normal gating modes that introduced a new open state. And the effect of this set of states was to cause the sodium channel to not inactivate completely. Even at depolarized membrane potentials, there remains some residual current through these sodium channels because they would enter this new set of states. They'd stay in this state, in this set of states for a fairly long amount of time, transitioning between non-conducting and open state, generating what's known as burst mode behavior. The channel would flicker open and close, and flicker open and close, and so on. Functionally, what this did is, unlike a normal sodium current, which undergoes complete inactivation in response to a depolarizing voltage steps, this is a negative inward current here, the voltage is maintained, it's probably something like 0 millivolts, or plus 10 millivolts, or 20 millivolts. Sodium current inactivates completely. In these mutated channels, there was some remaining sodium current that didn't inactivate, and that was due to the presence of this flickering burst mode that they had in the sodium channel model. So, different sodium currents, and they've got a mechanism for modeling it. That mechanism is based on electrophysiology done on this mutated channel. So they stick it into a cardiac action potential model, and they observe these things that I called early after depolarizations. And it's just a little blip here in this model, depending on when that depolarization occurs, the amplitude of the abnormal repolarization can be bigger. But the hypothesis here is that if this happened in enough cells at about the same time, this, even though it's a small blip, that could trigger an abnormally timed wave of propagation in the myocardium. And in fact, computational models of the whole heart show that this hypothesis has merit. This can happen. Enough with sufficient synchrony in enough cells, this kind of an event can trigger an abnormally timed wave that leads to a reentrant arrhythmia. And so the punchline here is that by using a model of the sodium channel, mutated sodium channel, they were able to take the nature of that mutation, stick it into a cell model, and understand something about the clinically relevant phenotype of the cardiac myocyte in the presence of this mutation. And so this kind of work has been done over and over in the field with many, many different types of mutations and relating molecular level events, abnormalities, to the integrative function of the myocyte as a whole. Now, this slide is intended to give you an appreciation for why that little blip, that EAD, occurs. So here's a simulation that we've done in another setting. It's not this delta KPQ mutation. It leads to an EAD with a very different morphology. It's a much more dramatic EAD. But I want to show this to illustrate one mechanism by which these events occur. And so here's a normal action potential recovering like so. Here is an abnormal action potential exhibiting one of these EADs. If we come to, I have to look at my screen because I can't even read what's on the slide, even from here. I don't know if you can possibly see it. So if we look down here at this trace, the y-axis is fraction of active L-type calcium channels. So a calcium channel is active if it is not voltage inactivated and if it is not calcium. If it's not either of those two things, it can gate. It can, well, it gates anyway, but it can open and conduct. And so this curve here, this dash curve right here, is showing the fraction of channels that are not undergoing calcium-dependent inactivation. And so what you see is that with action potential prolonged, so here's where the EAD begins to occur. That's the red vertical line. Right? So let's follow it down here. And what we see is that well before that happens, the fraction of channels that are not calcium inactivated is increasing. Right? This curve is rising. Why does that happen? It's happening because during this action potential, calcium is released into the cell and it's being extruded and it's being pumped back into SR. So the calcium transient is declining in amplitude during that action potential. As it declines in amplitude, we see that the fraction of calcium channels that are not calcium inactivated is increasing. There are now more of them available to open. And so when we prolong the action potential, we're enabling this to happen. We're giving a time window over which calcium in the cell can be pumped back into SR. The calcium level is dropping. And so the calcium channels are recovering from calcium inactivation. They're available to open. And if we prolong the action potential, the membrane potential is still depolarized. So that'll tend to drive the channels that are now becoming available to open into the open state. And that's what happens here. The calcium channels begin to rush into the open state, generating an inward calcium current that triggers that EAD. Now, I went through this, not just so that you could understand the mechanism, but so that you can see that this is a very general mechanism of arrhythmia. Anything that prolongs action potential duration, whether or not it's got anything to do with calcium channels or sodium channel mutations, anything that prolongs action potential duration has the possibility of contributing to this, to the generation of these kinds of arrhythmias, of cell life. And the list just goes on and on of all the things that you could change that could be perturbed in myosin that might make this happen. And so this is a very general mechanism by which cellular arrhythmias arise. And if this happens synchronously in a critical mass myocardium, that could trigger an arrhythmia. I will conclude with a model failure. There's all these clever sayings about the value of a model, and I wish I could repeat them word for word, but one is, all models are wrong, some are useful. I showed you a model we had that was useful in understanding heart failure, but I showed you it's wrong in many ways, and you can see one way in which it's wrong. And the other one is, sometimes we learn more from model failures than we learn from model successes. And so let's talk about that. So the kinds of models that we worked with early on were called common pool models. And this is what that means. In common pool models, we assume that the calcium that is released from the JSR is directed into the same calcium compartment that the trigger flux, that's my 10-minute warning, and I'm almost done, is directed to the same compartment as is the trigger flux. And that is the compartment that is driving the rheanidin receptors. That's the calcium concentration that's driving the rheanidin receptors. So that's what it means to be a common pool model. What does that imply? Now this is intuitive. Our mental models do work here. Imagine that calcium channels, the membrane potential is depolarized. Calcium channels start to open, calcium starts to flow into this common pool, the calcium concentration grows in the common pool, the rheanidin receptors are sensing that calcium concentration, they bind calcium, they begin to open, they release more calcium into that same pool. What's going to happen? This is strong positive feedback, regenerative feedback. Once release begins, it's going to promote release. And so common pool models have a property that is known as all or none release. Once calcium release starts, it escapes control. Boom, it just happens. It's going to start at a particular threshold voltage, and it's going to explode, and you can't control it, and the JSR just dumps its calcium. So this is how a common pool, any common pool model would behave. This is membrane potential, so we're varying membrane potential to recruit different fractions of opening calcium channels. We're using this to grade the trigger flux that drives calcium release. This axis is plotting the total calcium released in the model. The yellow is the model. So you see there's a particular membrane potential at which the trigger flux is big enough to cause rheanidin receptors to start to open. And the minute they start to open, the millisecond they start to open, calcium concentration goes up, and it's a positive feedback effect, and boom, you just get complete release. And so you have a mode where there's no release, a mode where there's no release at high membrane potentials, and a mode where it's just complete. But if you make this measurement in real cells, if you actually measure the trigger flux, and you measure the release flux, which can be done, instead you find it's a nice, smooth, graded function. It doesn't exhibit this all or none release property. We knew that. We knew the model was wrong in this way. And yet the model seemed useful, right? So all models are wrong, some seem useful. Our model seemed useful, and so we scratched our heads and for years couldn't understand, who cares? If the models are doing a good job and are so predictive, and both of the models that I showed you here are common pool models that are predicting useful things. Is it not important that in the real system, release is graded in the way that I'm showing you here? We couldn't understand. We thought it wasn't important. And then we realized that it was. We suddenly understood what was missing in the models, what was wrong about the models, and it had to do with this calcium-dependent inactivation. It goes back to that discovery that Dave, you contributed so much to. And once we realized there was an aspect of calcium-dependent inactivation that was not being correctly described by the models, and we put it in there, they fell apart completely. The models could no longer even reconstruct the simplest thing in the world, the action potential. Remember I showed you in the first lecture, these simple, early 1960 models that did a great job at reconstructing the form of the action potential, and they did it in the wrong ways. But it was easy to do. Well, we couldn't even generate a stable action potential. So we'll talk about this next time. It turned out to be an extremely important model failure for us because it's guided everything we've done since then and what many other people have done in modeling for cardiac monocytes. So we'll pick it up there.